All right, everybody, welcome back. Welcome back for this last session of our first day. Hopefully everybody's been enjoying the day so far, and we'll wrap it up with a really cool talk on plant science results. Our chair for this session is Liz Warren. She's Senior Program Director at CASIS, otherwise known as ISS National Labs. Hi, Liz, welcome. So good to have you here. And without further ado, here is the agenda for this session. So I'll let you take a quick look at that. We've got some really pretty cool stuff going on there. Plants and stress, I think I can relate to that. Okay, so with that, let's get right into our session. So let's roll the first one.
We are pleased to present our Healthy Hydroponics project for phytobiome management under controlled environment conditions. This project is funded by OMAFRA, the Ontario Ministry of Agriculture, Food and Rural Affairs, and the NRC Industrial Research Assistance Program in partnership with Ontario Greenhouse Vegetable Growers and International Phytobiomes Alliance. First, a few words about the concept of the We are a microbiome analysis company. We provide DNA sequencing and bioinformatics analysis of the microbiome. This includes the various environmental and agricultural sectors, as well as we develop products and services, including microbial inoculants, DNA extraction and sample collection kits, and methods for detection of pathogens in hydroponic systems. Our mission is to enhance food safety and improve prediction and mitigation of crop diseases in hydroponic systems with a circular bioeconomy context. The concept of the phytobiome includes all organisms associated with a crop plant and their interactions. This includes the various microbes and microbial communities, as well as arthropods and other animals. The interactions are influenced by environmental parameters and management practices. Within controlled environment systems, variations in environmental parameters are minimized compared to field-based agriculture. Our research on Earth is in the context of circular bioeconomy. We are developing the ability to couple food waste and bioplastics production in cradle-to-cradle cycles, with controlled environment vegetable production, shown in the circle on the left, playing an important role in using food waste-derived fertilizer, water, and energy to produce healthy plants in an environmentally sustainable and commercially viable manner. Controlled environment agriculture for vegetable production is done in several different formats using nutrient solution that is distributed in various ways, and different combinations of natural and artificial light. Shown in this slide are a vertical farm using energy-efficient LED lights, a fully hydroponic system with the roots directly in water, a highly productive vegetable greenhouse, and aeroponic system in which nutrients are delivered to the roots by misting or spraying, aquaponics, which combines both fish and plant production, and container farms. Also shown here is NASA's Veggie system, which has produced vegetables on the ISS. Building on our earlier work, we have started a new project. We want to get a better idea of what the microbiome looks like in hydroponic systems. So in the Healthy Hydroponics project, the goal is to refine and implement a rapid microbial monitoring system that is dependent on DNA sequencing and carry this out in greenhouse hydroponic environments. The system would be able to monitor baseline microbiome and also to track crop disease-causing microorganisms and food-borne human pathogens. The outcome of the project is to increase food safety through early detection, thereby reducing food waste and also improving human health. To reliably produce healthy, nutritious food on the ISS under controlled environment conditions, it will be necessary to be able to control the microbiome for maximum benefit. This will require building on what we learn from our experiences with and study of controlled environment cultivation in indoor farms and greenhouses. Fortunately, the tools are available, but there is still a substantial gap in knowledge as those tools have not yet been applied to perform the necessary research. Phytobiome management can be a core component of sustainable food production, food security, and food safety on Earth and beyond. If you're interested in finding out more about our project, please contact us on our website, healthyhydroponics.ca. Send us an email or call us. Also, follow us on social media. Thank you for your attention.
Hello, I'm Dr. Richard Barker. I work at the UW-Madison Botany Department in the Gilroy Astrobotany Research Lab. For many years, we've been collaborating with NASA on a project known as TOAST. TOAST is Test of Arabidopsis Space Transcriptome. In this study, we have been investigating how the model plant Arabidopsis responds to the stimuli associated with the spaceflight environment. As part of the latest installment of this program, known as NASA APEX 05, that stands for the Advanced Plant Experiment 5, we had the opportunity to grow Arabidopsis in the veggie growth system, alongside a mutant which has a reduced capacity to produce reactive oxygen species. So here you can see the hero of our story, Arabidopsis thaliana, growing on a square petri dish in the veggie growth system at the Kennedy Space Center. Its roots go down towards the gravity vector, and its shoots grow up and open to optimize their light harvesting capacity so that they can photosynthesize and produce sugars to fuel this growth. So these two distinct organs over time have evolved to have very different roles in plant growth, and so sense and respond differently to the different environmental stimuli that are around them. So during this experiment, we'll be harvesting the roots and shoots separately and discussing their function and responses separately. So here are those plants growing on Earth and on orbit. The astronauts, Scott Tingle, did a tremendous job taking photos of the plants as they grew. On Earth, you can see the roots going down towards the gravity vector and away from light. And here on the International Space Station, you can see the roots doing these fascinating things like curling off or just going upside down. It's awesome. It's just as we imagined it to be, and more. And then we got to watch the mutant plants grow as well. And here you can see the ROS mutant. So these plants have the reduced capacity to produce ROS. And as you can see, the plants grew really quite well. Like, we were really surprised when we saw these photos coming back down from the space station. At the end of the experiment, we got a lot more photography, and we were able to some of these images and find some patterns that we could do statistics on. And essentially that we saw that the wild type, its leaf area, its shoots, were significantly smaller in orbit. And we just didn't see that in the mutant variety. ROD, its shoots were the same size on Earth as they were in space, and this was just awesome to see. We also examined the root system architecture. And so as the roots grew on Earth, we really couldn't see a difference in speed. So this is time along the x-axis. But what you can see when we go into orbit, the mutant variety, its roots grew faster than the wild type. And that was really exciting to see. So it appears that reducing the ROS production in the apoplast of the plant appears to enable them to grow faster in the spaceflight environment and allows them to maintain the shoot size that we would predict based on their growth on the plate. So essentially, the RNA-seq data that we have is a blueprint for the plant's cellular response to spaceflight. And so we can start to see how all the different parts of machinery are changing to help the plants grow and survive in the alien environment of the International Space Station. So we're just going to focus on the root response to spaceflight and see how the wild type and ROD roots differ from one another. If we look at the bottom of this heat map, we can see two main GO terms that are distinct to both the wild type and mutant. You can see here in the wild type, it has a distinctly different amino acid transport system that is being altered by spaceflight, and ARBO-D has a distinctly different cellular amino acid metabolic process that's being altered. So essentially, the roots are dealing with their amino acids in a different way. And this is really fascinating. So this is fascinating because for a while, our lab has been researching the long-distance systemic signaling system in plants. And one of the things that we realized is if you add amino acids to the plant, apoplast, essentially you can create these really quite dramatic long-distance waves. And this begs the question, how is this long-distance signaling system affected by the stimuli of the spaceflight environment? So finally, it just gives me the opportunity to thank my colleagues at the UW-Madison. This work wouldn't be possible if it wasn't for Dr. Arko Bakshi, Dr. Richard Hillary, Dr. Wongyu Choi, and Dr. Sarah Swanson, and of course, Professor Simon Gilroy.
Hello, my name is Michael Wilkinson. I'm the Director of Education for Magnitude.io. I'm also the principal investigator for our research in nitrogen fixation in microgravity. Magnitude.io has a distributed model for student space science, with classrooms around the country acting as co-investigators, collecting control data with an analog of the flight experiment. Focusing on nitrogen fixation comes from my childhood in Iowa, where crop rotation of soybeans, alfalfa, and corn was the norm. I was curious as a child why that particular sequence and why that set of crops. So very early on, I was aware of root nodulation of legumes. And that early interest has developed into seeking a sustainable way to produce food to support space exploration. Synthetic fertilizers are far from sustainable in the space flight environment in terms of safety, energy, and upmass. Therefore, a biological solution is required.
Hello, I'm co-PI Lori Waters. Nitrogen fixation occurs when soil bacteria convert atmospheric nitrogen into nitrates that can be metabolized by plants and brought into the food chain. However, under stress, legume plants often prevent infection of their roots by the bacteria. Therefore, we are investigating how the stresses of microgravity, spaceflight, high ambient temperatures, and an enriched carbon dioxide environment, like those found on the ISS, affect the symbiotic relationship between rhizobia bacteria and legumes.
This is Gil Cawthorn. I'm a co-investigator at Magnitude.io. If legumes can successfully nodulate in the spaceflight environment, food crops can be grown without the need of nitrogen fertilizers. Not only could foods be produced from the legume crops, but by enriching the growth media with dead plants after harvest, growth media can be developed that would support nitrogen-intensive crops like fruits and grains. From this cycle, future crews on the moon or Mars could create a soil building process that would convert regolith to viable soil. ExoLab 8 is scheduled to launch aboard NG-15 in February 2021. While ExoLab is in orbit, we seek to achieve nodulation of clover seedlings. Several legume-based experiments have been conducted on the ISS, shuttle, and uncrewed missions by ESA and Roscosmos. None have achieved nodulation. ExoLab 8 seeks to identify and mitigate factors that may inhibit nodulation. We have identified two critical points of nodulation interference that will be addressed in this flight. We will first attempt to reduce the stress of launch by slowing germination through seed variety selection and pre-germination protocols, including manipulation of light exposure and temperature. This experiment's launch protocol seeks to mitigate the stress responses that produce salicylic acid and interfere with nod factors signaling of the flight specimens. The anti-inflammatory properties of salicylic acid may inhibit nodulation while also interfering with nod factors associated with the conversion of the pathogenic rhizobial infections to a symbiotic state.
Applying what we have learned in the past 2 missions, Exolab 8 will fly red clover seeds in a nutrient-free growth media that has been inoculated with rhizobia. Clover was chosen for its small size and reliable nodulation between 18 and 28 days in ground trials.
Exolab 8 will utilize the microgravity environment of the U.S. National Laboratory on the International Space Station to observe the processes of plant growth and development of root nodulation of legumes in a spaceflight environment. Previous experiments have not been specifically designed to investigate the nodulation process in microgravity, making the science goals of this mission unique. Continued research in the effects of root development in response to microgravity will further the knowledge and understanding required to develop plant strains and growth strategies that will address the challenges of nutrient and water uptake, both for spaceflight environment and agricultural applications here on Earth.
Hello everyone, my name is Arkadipta Bakshi. I'm a research associate at UW-Madison. Now I'm going to give you a snippet of my research on plants and stress, a journey from Earth to space. Do you know that by the time I'm done with this 5-minute talk, approximately 75 people will die due to hunger or hunger-related causes? The current world population is 7.4 billion and estimated to be 9 to 10 billion by 2050. According to the World Food Programme, an increase of 70% in crop yield is required to feed that population. Currently, that seems to be a huge challenge due to all the stressful climatic conditions such as flooding, which in turn lead to drastic reduction in crop yield. Hence, we need to devise smart ways to make the crops more tolerant to such extreme climatic conditions. Now, how can we as plant biologists tackle this problem? When plants encounter stressful conditions like flooding, they elicit calcium waves in response to it, as depicted by these green lines on the leaves. In this particular study, we have focused on the role of the TOUCH2 or CML24 gene, which acts as a potential hub in stress-responsive calcium signaling networks. In this research, we have used 2 varieties of the model plant Arabidopsis, which belongs to the mustard and cabbage family. One is wild type, where the TOUCH2 or CML24 gene is switched on, and the other is a mutant plant where this gene is switched off. Then we have studied the effect of flooding on these plants. Post-flooding, wild type plants are found to be highly affected compared to the TOUCH2 mutant plants. Pigment formation is another classical example of stress response. As fall approaches, gradual change in temperature leads to change in color of the leaves. The green color indicates chlorophyll and the red purplish color indicates anthocyanin pigments. From our studies, we found that the wild-type plants produce more anthocyanin, whereas the TOUCH2 mutant plants retain their chlorophyll, showing tolerance towards flooding. Now, using molecular biology techniques, we found that genes responsive to flooding are highly expressed in wild-type plants as compared to the TOUCH2 or CML24 mutant plants. So in a nutshell, plants with TOUCH2 gene switched off are found to be more resistant towards flooding, where wild-type plants were more sensitive under flooding conditions. Now we will switch our gear and let's go to with increasing climatic changes and population explosion on Earth, we need to find an alternative home planet for our survival as well as to save Mother Earth. 50 years back, the Apollo 11 spaceflight mission first landed humans on the moon, and now scientists are aiming to have a permanent moon base and eventually colonization on Mars. To support that, we as plant biologists need to know how to grow plants in outer space. But we have to keep in mind that in space, the behavior of water is driven by surface tension and not by gravitational pull. and it moves in all directions, creating this sticky environment. So when we will water the plants growing in space will be covered with water, and that will create or simulate this flooding like condition, which is why we had sent wild type and touch2 mutant seeds to the International Space Station via SpaceX mission, where the astronauts germinated and grew those seeds in their own space garden, also known as the brick unit. After the course of the experiment, the grown plants were sent back to Earth and eventually to our lab. Using similar molecular biochemical assays, we found that the wild type plants experienced a flooding like stress conditions in response to outer space microgravity, whereas the touch2 mutant plants were comparatively tolerant. If you want to find more, please listen to my full talk for more findings. Now, just to wrap up, a food for your thought. So if the touch2 gene is switched off and can be bioengineered into food crops, then the crops in question will be well equipped to combat such stress conditions on Earth and possibly in outer space. We hope our research focused on touch2 will be a great tool to bridge this gap, taking us into a greener and agroeconomically stronger tomorrow. So far, we have achieved several milestones with the advancement of science and technology. Although currently it might seem like a dream to have a permanent human base in outer space, but we are very hopeful to make this dream a reality with the power of science. Thank you. Hello everyone, my name is Arkadipta Bakshi. I'm a research associate at UW-Madison. Now I'm going to give you a snippet of my research on plants and stress, a journey from Earth.
Okay, everybody, just letting you know, this particular presentation has no audio with it.
We are all high school senior students at the Craft Academy for Excellence in Science and Mathematics at Morehead State University, conducting plant biology and microgravity research. I'm Allie Lewis.
I'm Jay Joshi.
Hello, I'm Grace Stubblefield.
Hi everyone, I'm Maiqi Chen, and we are the ExoLab leadership team.
Today, we'll first overview the background of our research project and our partnerships. Then we'll explore the different aspects of our actual experiment and delve deeper into the relevance and impact our research could potentially have on creating a brighter future on Earth.
The Craft Academy for Excellence in Science and Mathematics is the dual enrollment program we attend. The Craft Academy has a legacy in space. Craft Academy students have placed several experiments on board the International Space Station since our opening in 2015. This ExoLab experiment will be our 6th experiment in space.
The ExoLab research project is an international collaboration between the parent partner Magnitude.io out of Silicon Valley in California, the space-based experiment developer Space Tango out of Lexington, Kentucky, and all the participating students, schools, and organizations around the world. Our research is fundamentally a plant growth experiment that takes place in classrooms, at home, and on the International Space Station, where we run control experiments here on Earth in the Academy and compare the results to the experiments conducted aboard the ISS.
The experiment we are currently participating in is ExoLab 8. ExoLab 8 looks at the legumes and their ability to convert nitrogen from the atmosphere into molecules that we can eat through the process of nitrogen fixation. Legumes have a symbiotic relationship with a species of nitrogen-fixing bacteria called rhizobium. The bacteria will be incorporated into the growth medium of the legumes and be launched to the ISS in February of 2021. The novel goal of ExoLab 8 is to achieve root nodulation of legumes in microgravity, something that has never been accomplished before.
Before Mission 8, however, Magnitude.io had 9 potential legume candidates for launch. Over the summer, we received supplies from Magnitude.io to perform our own flight legume determination experiments at home, along with other students across the world. Recently, red clover was declared as the legume of choice to fly aboard the ISS due to its size and hardiness.
The red clover seeds will be sent to grow for 30 days in microgravity, caught in Earth's orbit aboard the International Space Station. Here on Earth, we will have near real-time imagery of the growing red clover seedlings, observing how microgravity impedes or improves growth, and whether the nodulation that occurs on Earth through the symbiotic legume-rhizobium relationship can be replicated in space.
As mentioned, when the experiment is launched and conducted aboard the ISS, we will run the same experiment here on Earth. By doing this, we can gather accurate real-time data from the modules on Earth, from our lab, and from classrooms all around the planet to compare to the ISS experiment. Once our experiment has returned to Earth, we will analyze the samples to better understand how a microgravity environment affects the chemical process of nitrogen fixation. Nitrogen fixation is an essential process of agricultural systems and a necessary component for translating agriculture to spaceflight environments. The result of this study will further our understanding of how specific strains of bacteria can aid in the successful growth of crops in space.
Our purpose of conducting this research is directly relevant to the United Nations goal to solve the world hunger crisis and soil depletion. The nitrogen-fixing characteristic legumes possess lend to enriching the soil for other non-nitrogen-fixing plants to be able to thrive in once-depleted environments. Not only do legumes enrich soil for other plants, but they also emit less greenhouse gases, are more storage-friendly, therefore reducing food waste, and offer multitudes of nutritional benefits compared to other contenders for solving this world issue. Our involvement in ExoLab 8 will help scientists better understand the nitrogen-fixing process and potentially serve as part of a larger world hunger solution.
Arkadipta Bakshi. I'm a research associate at UW-Madison.
Hello, my name is
Now I'm going to
anamika.ray
give
and
you a
I
snippet
work
of
for
my research
InnoSense
on
LLC.
plants and stress,
We
a
are
journey from Earth to
prime
space.
Okay, everybody just stand by 1 second. We just think we had two presentations going at once there. So hang on just 1 second. Be right with you.
Hello, my name is anamika.ray and I work for InnoSense LLC. We are the prime in this project, and the project was sponsored by NASA Kennedy Space Center. The title is a portable and ultrasensitive ethylene sensor for real-time monitoring in plant growth habitat. This is the executive summary with an abstract ID 337, and the subcontractor on this project was Arbitech. The contact information is provided here, and my contact information is also provided. If any one of you have any questions, please feel free to contact us. So we developed an ethylene monitor for NASA. NASA did not have a real-time ethylene monitor for plant growth habitat. So this is based out of ISL's proprietary microelectronic device sensor platform. This because of polymer nanowires grown on the sensor platform, which are really tiny sensor chips, silicon-based sensor chips. It provides very high sensitivity and stability and specificity with low maintenance. The customized recognition molecules of the copper complex here, which was used for detection of ethylene. So you see the gold pads where the polymer nanowire breaches the 2-micron gap and seals the circuit, and then it is copper complexes are grafted on the polymer nanowire. The copper complexes serve as receptors for detection of ethylene. So the detection of ethylene is selective, reversible, and fast. We also designed special gas lines for sampling and doing the baseline studies, which provided stable baseline and minimal use of consumables. The figure on the left is an envisioned prototype, and this particular sensor can be modified for use in various different kinds of plant growth chambers. It has a miniaturized system architecture and hardware design, which enables it to be which enables it to function under low power consumption and its minimal size and weight. So this is the prototype that we constructed, as you can see in the left-hand side picture. And the prototype was tested first at ISL, of course, and then again in Orbitech. It was shipped to Orbitech and tested there. So you see the testing here. The picture is of the testing which took place in Orbitech. And on the right-hand side is a bar graph showing the sensor response. So we saw distinct concentration-dependent response of our sensors. There were multiple rounds on the same sensor which was used, and we saw reproducible responses upon multiple exposures. And the dose-dependent response was seen where the minimal response was at 25 ppb of ethylene exposure. So this data provided very good confidence in our sensor system that the sensors are robust and reversible. So the accomplishments in phase 1 and phase 2 of the project, we fine-tuned and scaled up the sensor fabrication process, resulted in sensors that are reversible and sensitive to ethylene down to 25 ppb. Of course, the sensor results were validated by GC. We designed and constructed a standalone prototype, and also which was suitable for integration into the APH unit. And the design features include multi-channel data acquisition, automatic sampling, and we had scrubbing cartridges for scrubbing specifically various different VOCs, which also carbon dioxide, and also scrubbing the ethylene to bringing it back to baseline. We designed and developed a customized graphical user interface that provides data collection and display, which can be done very easily. And we independently validated the prototype performance at Orbitech. This independent evaluation showed consistent response of our sensors from 25 to 100 ppb ethylene. At our end, of course, we had done up to 1000 ppm of ethylene, and the sensors still showed reproducibility and reversibility. Orbitech was also overall satisfied with our overall sensor performance, concept, and operational capability of the prototype. Of course, this project would not have been possible without NASA's funding. Dr. Orlando Melendez and Dr. Raymond Wheeler, and of course the NASA consultant, Dr. Oscar Monge. Dr. Wheeler and Melendez helped us from the very beginning with detailed guidance and discussions. The Orbitech team, Dr. Bob Morrow and Mike Brennison, they provided valuable insights, and especially during such trying times with COVID-19, they did an awesome job in testing our prototype. And Jeff Paul and John Watkins for developing the prototype. With that, I would like to end this executive summary. If any one of you have any questions, please feel free to contact me. Thank you.
Excellent. All right, very cool presentations. With that, Liz, welcome. Good to see you as always. I wish it was in person once again, but good to have you here, and thank you so much for running this session. We've got some really good questions coming in online on the Q&A chat, the Q&A tool. So submit your questions there. And with that, Liz, I will turn it over to you to take it away.
Thanks very much, Jim. So hello, welcome. Thanks everyone for sticking with us at the end of our session today. We've got plenty of time for questions and answers, so I don't want to rush this. Please feel free to enter your questions in the Q&A window. As Jim introduced me, my name is Liz Warren. I am a Senior Program Director with the Center for the Advancement of Science and Space, CASIS, and we are very proud to be managers of the ISS National Laboratory. Personally, I've been involved in spaceflight research. I recently did the math and kind of blew myself away for almost 30 years now, and that's 30. But it's always exciting, and these presentations today were fantastic. Everyone is adapting to the new kind of virtual format, and I couldn't be more pleased. So as we approach 20 years of continuous human habitation on our International Space Station, I want to remind everyone, if it's not blatantly obvious, that microgravity, the spaceflight environment, has profound effects on living systems, from cells, microbes, animals, humans, and of course, plants. We've been growing plants in space for decades. We've learned a lot. Boy, we sure had some tough lessons early on, but now we're able to even grow food for astronauts. We've got a lot more to learn though, and a long way to go. And so some of the research that we heard about today are some of those steps and some of those next steps. And what is really exciting to me is that we've got young people through STEM programs involved in finding solutions. This innovative research that is ongoing is extremely exciting. And without further ado, I want to get to our speakers today. I'm going to briefly introduce them all, and then we'll get to the Q&A. So we first heard from Dr. Trevor Charles. He's a co-founder and chief science officer for Metagenom Bio Life Science Incorporated and director of Waterloo Science for Microbial Research at the University of Waterloo. He's a bacterial molecular geneticist aiming to use synthetic biology, genome engineering, and microbial community analysis in applications related to circular bioeconomy. And one note, so sorry that slide 2 had a little audio issue, so apologies for that, Dr. Charles. Next, we heard from Dr. Richard Barker from Simon Gilroy's lab at the University of Wisconsin at Madison. Dr. Barker has contributed to 3 or more successful spaceflight research projects, and he is co-chair of the NASA Plants Advanced Working Group and a member of numerous other NASA Gene Lab collaborative working groups. Next, we heard from Michael Wilkinson. He's the Director of Education at Magnitude.io. Michael is a mathematics and science educator and a curriculum and professional development consultant. Michael teaches 4th and 5th grade mathematics and science in the Bronx. He's Director of Education for Magnitude.io and the PI for Exolab 8, which we heard about today. Dr. Arkadipta Bakshi, a research associate also in Dr. Simon Gilroy's lab at the University of Wisconsin in Madison. Dr. Bakshi is working on several space biology projects to help plants to grow better in the microgravity environment, overcoming different environmental factors, primarily oxidative and hypoxic stresses. From the Kraft Academy for Excellence in Science, we heard from Maiqi Chen and a number of her colleagues. She's a high school senior in mathematics in Moorhead, Kentucky. I'm sorry, I just screwed up the title of your high school. She's currently enrolled in STEM and business college courses and has an interest in exomedicine. Upon graduation, Maiqi plans to attend college and pursue a career in neuroscience and psychology. Very exciting. And then finally, we heard from Dr. Anamika.ray from InnoSense, LLC in Torrance, California. Dr. Ray has a PhD in biochemistry and has extensive experience with nanomaterials-based sensors, characterizing and optimizing the sensors for gas sensing and in vitro diagnostics. So welcome all of you. Thank you so much for being here, and we'll dive into your questions. I've got a couple that I've already put together, but I want to get right to one of, let's see, I want to jump right to the STEM, to Kraft Academy and Maiqi, just because I'm so thrilled with what you're about to embark upon. Great project. We want to know, what criteria did you use to down select to get to your legume?
Hi, so thank you so much for congratulating us. We're also very excited about our research project. So as to the criteria, our affiliate, which is Magnitude.io, and you've heard from them earlier too, and their contributing scientists, they actually decided on down selecting the legumes themselves, and they picked red clover as the flight candidate. And some of the criteria that they used was the size of the legume, because as we found in our summer trials, some of the legumes grew super tall, so they outgrew the test tube constraints, which wouldn't be good on the actual mission. And they also looked at the timely development of the root nodules. And lastly, they looked at the robustness of the plants themselves.
Very good, thank you. Actually, I had a follow-up question for you since I've got you and since all of your teammates are there. Hello, everyone. Hello. So actually, I would love it if each of you would give us an answer to this question. First of all, you guys are light years ahead of where I was when I was in high school. I just dreamed of doing spaceflight research when I was in high school, so kudos to you. With regard to addressing UN sustainability goals and perhaps solving or bringing some solutions to feeding the planet, or maybe other UN sustainability goals, what would you think would be like a next step after your experiment flies? So, you know, tell us what you think you're going to find and then how that might apply back here on Earth. So first of all, each of you, I'd love to hear from each of you. We know what Maiqi is interested in doing after high school, but I'd love to hear from each of you.
Yeah, I can start. My name is Allie Lewis. As I'm sure you all know, I'm a student at the Craft Academy, and post-college or post-Craft Academy, I'd like to go on to college and pursue a career in the medical field. And I'd really like to see in this experiment, we're looking for the development of root nodules, and I really hope that we see this in this experiment because, as we mentioned earlier, it's a novel thing that's never happened before. So just in that in itself would be fantastic to see. So I would love to see that, and just the impact that that could have here on us on Earth would be amazing.
Mm-hmm. And before you move on, Allie, what do you want to do after high school? What goals do you have personally?
I'd like to go into the medical field, like I said, so just continue with that goal and hopefully continue to do XLab missions as well, because this has been such a blast. I've had such a good time just participating in this.
I'm Grace Stubblefield. My future goals are to attend college and become a genetic counselor. So hopefully I'll go to school somewhere, maybe Cincinnati, something like that. And then in relation to the UN Sustainable Development Goal, one thing we talked about and we learned a lot about was that this nitrogen fixation process and the nodulation of the roots of the legumes can actually be further used to replenish depleted environments. Depleted soil and depleted environments. So hopefully those areas where they have a lot of difficulty growing crops, we could move into those areas and try and build that back up again.
So much like Allie, I also kind of want to go into the medical field, but I'm also strongly interested in biomedical research. So I guess some of the major applications of this research would likely be, as Grace mentioned before, bringing back nitrogen into the previously depleted soil. But also it's very important to notice that it can also reinvigorate unusable soil, unarable soil. So that's another very important application that will bring more arable land to, I guess, more arid climates.
Fantastic. And while we're on the topic of Magnitude.io, Michael, you're doing fantastic work. What is next for Magnitude.io after NG15? Do you have further plans, and how do more students and classrooms get involved?
Well, the latter question is the easiest one. Drop an email to hello@magnitude.io, and we'd be happy to have our co-investigators. Our students are really and truly co-investigators. They're not playing around. They're not mimicking the work. They are part of our science team. And as you know, Liz, as a scientist, you got to have a solid control base in your data to know if anything's actually happening. So that's really what they're responsible for. What's next? As I said in the piece, the whole legume rhizobial symbiotic relationship is one that's long been of interest to me. So it's really exciting to be able to be pursuing this in a very rigorous way. And I would really like to continue on this path of developing a soil succession route so that we can really explore Mars and the moon, and maybe, maybe some of our Craft Academy colleagues will be the medical team there. We'd be in good hands if that were the case, I know.
Thank you very much. And
I mean, let me just add, one of the great things that we've been able to do is get away from the one and done experiment that's so common in space flight. We're trying to stick to this path, starting with with 6 and just looking at the microbes, trying to grow the cowpeas in 7 and now the clover in 8.
Yeah, it's been very nice to see you guys building on knowledge and not just doing one-off experiments. So that's been very encouraging. I'm going to shift gears now. Thank you, Michael. To our first speaker, Dr. Charles. Trevor, I was interested by your slide on circular bioeconomy. Kind of a little bit of a common thread here. Now, certainly on exploration class missions, when we're moving on to Mars, sustainable spaceflight is going to be critical. If you can you imagine for me and talk me through what a circular bioprocess might look like with plants on a Mars mission?
Yeah, so I mean, our context, of course, is the work that we're doing on Earth. But the way that we are approaching it is tackling both the food waste problem and also the materials problem, in our case, plastics, in one. So you can sort of think of it as taking food waste, which is very expensive and difficult to get rid of, and using microbes to convert it to a feedstock that can then be used to produce, in our case, bioplastics, which, on Mars, in the Mars mission, could be used to produce material. But also out of that process, you can extract water and CO2 and nitrogen and phosphorus that can be used as fertilizer to grow additional plants. So that's where the circular nature comes in. So just like we're planning to do on Earth, this is something that could be incorporated into a Mars mission.
Very cool, excellent. And certainly, I didn't mean to downplay the importance of sustainable practices here on Earth. I was just intrigued of how might we take this beyond?
Of course.
Dr. Richard Barker, you work in Simon Gilroy's lab, pretty much a legend in microgravity research, plant research. I have 2 questions for you. First of all, briefly, tell us what's new in the lab. And then I was curious, what is the advantage of the plants growing in microgravity? Why were they growing their roots faster, do you think, in your findings?
Hi, nice to see you again. So what's new in the lab? Well, because of this whole COVID isolation, the lab's gone distributed, and that's actually how we met through the Magnitude program. So we've been trying to write up a lot of the protocols that we use in our NASA experiments, and trying to share them with citizen scientists through social media to try and encourage everyone to collaborate and share data with this goal of going back to the moon, and not just to visit, but to make a sustainable community there. And that's why it's great to see such a wonderful community here, like of so many different generations. And the second point of the question was the not the wild type, but the mutant plants grew their roots faster in microgravity. Why? Yeah. What's the advantage there? Were they seeking nutrients? So the direction and the kinetics, yeah. So usually plants use gravity to provide a vector for the roots to grow in. And in the microgravity environment, that vector is missing. And so the question has often been, well, is that stressful? Do they need a vector? And if they don't have one, does that affect shoot growth? and not to mention the other factors that we think might be present. And so when we looked at all these things together, what we could see was that yes, the wild type plants had a smaller root system. They were being affected by the spaceflight environment and exhibiting a dwarfing effect that we associate with stress. And then there's been many papers in the past that have shown that the production of active oxygen species is a kind of key phenotype that transcends all species in the spaceflight environment. And so by removing the plant's capacity to produce some of that ROS, it just so happened that those mutant varieties grew much better. And it's intriguing to think what would happen if we could apply that technology using genome editing to microgreens and other like crops that astronauts and future space farmers might want to grow.
Someone else had asked, and you just touched on it when you mentioned citizen science, and I know you're a part of some Gene Lab working groups. Has there been a Gene Lab style omics investigation of plants in space?
In fact, there's been a few. So there are many different types of omics, and that's one of the wonderful things about Gene Lab. It's a very unique repository in the sense that it takes all types of omics data, no matter what species or what platform you're using. They really want to bring it all together and help annotate it, help make it accessible and interpretable. And we were lucky enough to be part of that program and help collect together all of the pioneering plant studies. And so we've collected together about 17 different transcriptional studies that are designed to investigate plant response to the spaceflight environment, but also different factors that we think could be associated with the spaceflight environment, such as radiation, such as like gamma radiation particularly, such as hypoxia and things that activate the heat shock system. Earlier on, we heard in the human cell part section, there was mention of like this chaperone system being activated as a defense system. And that is also true in plants. This heat shock response system is a system that involves the production of molecular chaperones that help proteins function in stressful situations, such as when there's changes in ionic balance in their environment because of too much salt or no gravity vector. So it's a very complex genetic network we're trying to unravel. And that again is the wonderful thing about the Gene Lab platform. It makes sure that all of the processing is all open and accessible at all the different stages. So bright young minds, students can come in and access the data at a range of different stages and do their own analysis and make their own discoveries. And that's essentially what the AWG community is all about, trying to bring these citizen scientists together, be it in high school, undergraduate, or non-associated with universities like beyond. It's a really cool program. Over.
Fantastic, fantastic. For Arkadipta, Bakshi, it is flooding right now in Houston. There's rain, but there's also storm surge from the Gulf of Mexico. So your presentation on plant survival when they're drenched or drowning in water was timely. Plants always amaze me with their robust redundant systems. If you could engineer a plant to go to Mars, specifically for food production purposes, like what would some of its traits be? I know this is kind of an off-the-wall question, but there's so much genetic engineering that you can put into a plant. What would some traits be that you would want?
Thank you, Liz, for such a wonderful introduction. And that's a wonderful question. I really care about the flooding related events. It's happening all over the world. It's one of the major events which occur every year. Coming back to your question, I personally feel like, so there are a lot of steps to go towards to make a genetically engineered crop, which would be flood resistant. Coming back to the research which I conducted or on the Touch2 based research, there are certain steps, as I mentioned, but tweaking those genes which can help the plant to grow better under flooding condition would be the first steps. And Touch2 would be a nice tool to tweak around. Obviously, we had done the research on the model plant Arabidopsis, which belongs to mustard and cabbage family, but Arabidopsis, that CML24 homologs you can find in a lot of almost all crops. So we need to translate that research into those food crops and then look at whether those crops are also sustaining or growing better in the flooded environment compared to the wild type plants where the gene is not tweaked. So I would start with that. And then obviously you have to look into the production for that particular crop. How is the fruit development, or everything is being affected with that tweaking of the gene.
Right, right. And then of course, you know, in regions that are not flooding, even though there's a great deal of flooding happening around the world, there's also drought, and drought resistance is also a critical solution, again, for crop production and feeding the people of this planet.
Could I interject there?
Yes.
So the next technology that we're testing on the International Space Station has been designed to address drought and salinity, which are the two major abiotic factors that are inhibiting global agriculture now and predicted to move forwards. So the technology the program is sponsored by CASIS and by Target, and essentially we've got these modified cotton plants that on Earth have already been shown to be resistant to these stresses, and we think they're going to alter the plant's response to the stresses of the spaceflight environment. So Arka mentioned we're pretty much focused on flooding for the spaceflight environment, but the technologies that we developed for that application, we know can be applied on Earth in a range of different situations, ideally in the future in underutilized crop species, as well as the elite cultivars. Over.
Thank you so much, and a great reminder of that upcoming research sponsored by Target Corporation, actually. Let's see, we're running a little bit short on time, so I want to make sure I ask a question to anamika.ray at InnoSense, who's developing that ethylene sensor. Let's see, the question was two things. What is the dimension and power requirements of your ethylene sensor? And then also, I was curious, you have an expertise in nanowires. Does humidity affect them over time, or what do you expect the lifetime to be?
So answering your question about the sensor dimensions, the sensors are really tiny chips around 3 millimeter to 6 millimeter. For power requirements, it's very low powered, battery powered, or also any kind of incorporation with APH or any other growth chambers used in space that can be done. Coming back to your question of humidity, yes, they are sensitive, and we kind of do some surface passivation, certain things, and we are working on that to even improve that.
And expected lifetime of those sensors on orbit?
So the expected lifetime studies that we have done, we have looked at like a couple of months, but we need to do more studies on that to see how long it would sustain in terms of reversibility and response to ethylene.
Wonderful. Okay, let's see. I'm looking through this list of questions. This is for anybody. For vegetables that are grown in space, this is kind of a two-parter. Certainly, we know from the veggie team and Joya Massa that there's psychological benefit to the astronauts of growing plants and caring for another living organism on orbit, let alone the wonderfulness of having fresh vegetables. But here's a question, and it kind of goes to a couple of these other developments. For vegetables grown in space, is there any concern that microgravity will produce unique and uncharacterized toxins or beneficial organisms as a result of microgravity? And I think that Trevor, maybe you might want to start with that.
That's a tough one. So I guess speaking on from the viewpoint of the microbes that are associated, one of the concerns that we do have is that there could be toxins or pathogens that are promoted during growth of the plant under those conditions. So because of that, it's really important to develop control mechanisms for the microbiome so that you're promoting a beneficial microbiome that is beneficial just not only for the plant, but also for the humans who are cultivating and then eating the crop.
Yeah, fair, wonderful. Well, actually, we are running out of time, that was the last question we're able to fit in. I encourage all of you listening, there are full presentations that each of these speakers has put together for you. I encourage you to go listen to the full presentation. You just got a little taste today. Thank you so much to our panelists. Very much appreciate your time and your effort. This has been a fantastic ISSR&D conference so far, and that is largely thanks to you. And Jim, I'll kick it back to you.
Fantastic. Thank you so much, Liz. And thank you to all the panelists, presenters. Great job. Fantastic stuff. We really appreciate you doing it. It was good to have you all here today. Thank you again so much, Liz. And hey, with that, we're done for the day. How about that? So thank you to everybody online for participating, for your great questions that you submitted during the day. And of course, thank you to our sponsors. Our platinum sponsor is Boeing, who's been the platinum sponsor of this event from the very beginning, back in 2012. And our silver sponsors, Jacobs, KBR, Leidos, Oceaneering, Teledyne Brown Engineering, and the Engineering and Innovative Technology Development Organization at the University of Alabama in Birmingham. So please do come back tomorrow for our second day of these excellent tech sessions. We start off really early at 1100 Universal or 0700 Eastern with an international focus on ISS utilization. There's a separate registration for tomorrow, so please go to issconference.org and get yourself set up to attend tomorrow as well as Thursday. Please do join us for Thursday. And with that, that's it from here, and we'll see you all tomorrow. Take care.